You've all seemed to identify that the B cell is a major culprit in lupus. So what I'm wondering is, in your studies of investigating the B cell autoantibody production at various stages along the way, did you find any clue as to what actually causes the B cell to become overreactive in the first place?
So the B cells do produce the autoantibodies. I mean, one theory where both atacicept and belimumab can potentially work is, at least in animal models, it's pretty clear that if you have elevated levels of BLyS, then the clones of plasma cells that produce autoantibodies are allowed to survive and escape the what's called apoptosis or programmed cell death. So what BLyS and APRIL can do is induce molecules that prevent the sort of digestion, the killing of the cell. So they actually inhibit the signal that says the cell should die by preventing enzymes called caspase enzymes that will auto-digest the cell. They actually inhibit all those enzymes. When the BLyS levels can go down, that signal is now allowed to go forth, and those cells can die. This is in animal models. In mice, it's sort of like BLyS can be a gatekeeper. If you have high levels, the autoimmune cells escape, go out into the circulation, produce autoantibodies. If the levels are low, they will undergo their programmed cell death. That's one theory.
He's sort of giving you a picture of the immune system as a circle, going around in a circle, and you're constantly needing to maintain a balance in your immune system, because you want it to be able to fight off invaders, but you don't want it to cause lupus, right? So as it goes around in its circle, cells constantly need to be born and cells constantly need to die in a very organized way. So that if you have a virus, some cells need to be born that'll fight off that virus. And then after that's over, you need to get rid of them because they're going to cause trouble, and they could cause lupus if they hang around. And what he's saying is that if you have too much BLyS, that circle's going to get kind of off kilter, and you're going to have too much of the immune activity, and you can't kind of get rid of all the cells that are causing all the trouble. So one of the ideas is, if you interrupt this, that the natural process that would kill off the cells can be more protected. Would that be correct?
Correct.
Yeah.
Only one of you mentioned NP lupus, and it was as an exclusion from a study. Does anybody study it? Do any of your drugs affect NP lupus or just nephritis?
One of the problems right now in drug development is that we don't have a worldwide connection where we can get enough patients with, you're talking about neuropsychiatric lupus, yeah, enough patients together to get a really good interventional trial going. I think that will change over the years, because one of the things I'd like to have them comment on here is the fact that everything's changing. Things are becoming much more global. Their development of their drugs is becoming more global. And I think when that happens, we're going to be able to see trials in more rare manifestations of lupus. But yes, somebody is studying it avidly. Dr. John Hanley of Halifax is doing a worldwide study of neuropsychiatric lupus, and he is going to come to them with all the data they need to be interested enough. Okay. So in 3 years, we'll give you an update. But I'd like some comments about that. Are you guys are going global, right? Oh. We used to talk about developing drugs in a country. We don't do that anymore.
You know, no one develops a drug in a country, and certainly no one could find enough patients to fulfill the requirements for a phase 3 study without doing global studies. So that's obvious. I think that, you know, if you take a step back about neuropsychiatric lupus, is really the key question. As with lupus nephritis, you can't really compare it to, you know, everybody gets standard of care, but you would have to give it against a significant immunosuppressive drug, as you do with lupus nephritis, because it's not appropriate not to do that. So in fact, it becomes very difficult in the studies to make that comparison when you're comparing it. You don't want to give a drug to somebody who can have very, very bad manifestations, yet not get a drug that may be helpful, although it has a lot of side effects. So studying neuropsychiatric lupus is quite difficult at this point because of some of the ethical issues. And the same thing with lupus nephritis. You have to kind of give them at least standard of care, something that's going to treat their nephritis. And sometimes when you're treating them with a drug that you don't know how effective it is, it's a little tricky. So it's not like we avoid neuropsychiatric lupus. It's just that the ethics and the combination drugs you have to give makes it a little more difficult. I don't know if anybody else wanted to make a comment like that.
So in our trial, what I said was we excluded people who had active severe. It didn't mean if someone had more chronic and stable that they couldn't come in. The concern is that the drug isn't going to be active immediately in anyone who has a potentially life-threatening or requires high-dose steroids or IV cytoxin, such as for vasculitis. They would be excluded. So some of the patients come in, but the other thing is in the trials, that doesn't mean that people can't develop new symptoms, and you're always analyzing that. And actually in our phase 2 trial, we showed that there was a reduction in the CNS by BILAG with that, because every 100% of the people have the ability, potentially may evolve into that. So it's not seeking to show the improvement, but you're always going to be looking at those who may worsen in the trial, because that's...
You mean the placebo group flared? There were
Yes.
more flares in the placebo group than
Correct.
in the treatment group, and that was possible because you went global. You did a huge trial.
Correct.
You had enough
Well, that was
patients
in,
so
that was in phase 2, what I'm
Oh,
talking
okay.
about.
Well, it was still a pretty big trial.
Yes, yes.
That wasn't a small trial, Bill.
But the point is, you're looking for the improvement. All the trials will always capture potential worsening or less worsening because those are new events. But the neurocognitive and those require special instruments to do that, and we didn't do that. And if you're going to do that, you really would need to do a specialized trial so you could apply it to 100%.
I think it will become more possible when we understand the biology better, and I think that's what Dr. Hanley's trying to contribute now. When we understand the biology better, there will be products that will just come along that will seem right, and they will have to be added. It will be problematic. It would have to be a worldwide trial. It would have to be added to standard of care. Any given trial site might be told you're going to enter 0 patients or 1 patient or 2 patients in a year, and that's all you're going to find who are going to qualify for this study. But I do believe someday this will happen.
My name is Eileen Fraser. I'm the provincial contact on Vancouver Island for the area called Oceanside, which is Parksville and Qualicum. I think I'm going to say the new drug wrong to this doctor. Is it prednisone sparing, the Pulumab? Yes.
So in the trials, we have seen what I would describe as prednisone-sparing activity. So depending on how you analyze the prednisone use, in some cases it is statistically significant, in others it isn't. When you look at the ability to reduce prednisone in the last 12 weeks in the study that I was showing you specific results of, there was a reduction in both doses that was either statistically significant or right at that level in reducing it compared to the placebo. But steroid sparing works both ways because you can have worsening, so you have to look at it by worsening, and when we looked at that, the higher dose did that and the lower dose didn't. We've actually looked at steroid sparing because there is no defined definition, multiple ways, about 10 different ways to understand it, and we've shown that there were a greater number of people that had a 50% reduction at any one time from 24 to 52 weeks in that one study. So there is, in general, patients are able to use less steroids over time on belimumab. Some people in our long phase 2 have gotten off steroids altogether. Others, have not. So that is one thing in addressing the question before of getting off all the medications. It is unfortunately sort of one step at a time. In our phase 3 trials, we did not mandate a steroid taper. It was clinical judgment, and in a blinded fashion, the clinical judgment suggested those people on belimumab used less steroids and had less increases. Ultimate goal is to get off. That certainly wasn't significant. So we look at that. You look at ability for immunosuppressive agents and all of that. But that wasn't, we measured it, but the trial wasn't designed to show steroid sparing, though it did show that there was some activity in that direction.
You know, I'd like to find out how important steroid sparing is to this group. So if you had a choice to stop your Imuran or to stop your steroids, who would stop the Imuran?
I would stop the steroids.
Who would stop the steroids?
Absolutely.
I think that was informative. Thank you.
It's Kirsten from Lupus Europe. It's really fantastic to hear what you are telling us today, because it's a completely different message than from just 2 years ago. So congratulations on that. We appreciate it a lot. One thing that we have learned just since the past year is to redefine some of the results, some of the findings. And I was wondering if there is any way that we can organize that we, the patients, can help redefining some of the results that the clinical trials will be showing.
Talk about patient outcomes, patient-reported outcomes. You have some data, right? Are you allowed to say some stuff? Okay. You have.
So you're talking about patient-reported outcomes. So we have reported at the ACR, American College of Rheumatology, that slide I showed you on fatigue, and then quality of life as measured by SF-36 was not significant at 24 weeks, but was significantly reduced at 52 weeks. And then we have a poster here about quality of life in that first study, and we show the breakdown of the different components of SF-36. So there's one physical domain and mental domain, and what we show in there is there's one of the domains called vitality, which is where all the fatigue questions are, and you get a response very similar to what you get with the FACIT fatigue. So as a way of validating that it's real in the timing and that, and then multiple other, 3 other domains in the physical component score were significantly reduced in that trial in the 10 mg per kg and some doses of the 1 mg per kg. So we have reported that, and there is a poster on that on Saturday.
And that has helped a lot, yes. But I was thinking further on, perhaps, also to include some of the other manifestations that that could be difficult to handle, perhaps.
So what are you? Go ahead.
We have a response to that, actually.
Okay.
Yeah.
What do you mean? I guess, so I was going to answer the first part of the question, but the second part I'm a little confused about, so if you may be able to help you. The other manifestations that are difficult to I'm not sure.
Relationships, work.
Exactly.
Okay,
That's what we got.
okay,
Or
fair enough.
mental state.
Okay,
you know.
so, you know, I think that unfortunately, what we end up talking about are measures that are not as meaningful to the patient. But quality of life measures are important for everybody on this podium. So I think that everyone in their trials are doing multiple quality of life measures. I think they just mentioned the SF-36, the FACIT fatigue. We have a survey that we're presenting to look at what manifestations of a concern, and certainly it's your work productivity, it's your ability to do your work in your house, it's your ability to deal with your sexual activity, it's your ability to handle your kids and don't need help in the house. There's a whole series of other manifestations that people are worried about, and we have done a survey and we are going to present. It has nothing to do with the drug, and want to know what's important to the patient. And when you know what's important to patients, you then can look to see if the drugs make a difference. And we know how important it is for you. It's very important for you to want to be a productive member of society, to be able to do everything you want with your family, to not feel fatigued, et cetera. So we know those are key manifestations for every patient, and we're worried about the patient. I think that for the regulators, it's sometimes easier to measure these scales that probably don't mean as much to you.
Hi, I'm Susan Milliken, and I'm from the BC Lupus Society, and I've been going to the annual symposiums that have been put on for several years. And a few years ago, a renowned rheumatologist came and spoke to us, and he told us that there were some design problems with some of the drug trials, and that some very good drugs had been eliminated for that reason.
Had been what?
Sorry?
Had been what?
Eliminated.
Eliminated, yeah.
And I'm wondering if you have been able to find a way to bring these drugs back to being considered for...
These guys spend half their lives doing that.
I mean, first of all, I could say that my colleague over here is the only 2 phase 3 trials that have been positive in lupus, so obviously they bypassed that issue. As far as the drugs that we didn't develop, we have to go back to the companies that did that and ask them if they're going to do, try to bring them back if we think there's activity. But I think that they have overcome that issue, and if you look at their posters, you will see they have 2 positive trials. I mean,
We're struggling with these trial design issues still, but we finally are beginning to see a light at the end of the tunnel, and thanks in large part to some of the people at this table. So yes, I agree with you. I hope there will be a way to revisit certain agents, especially some of the ones that are being used in lupus.
Right. Well, thank you very much for all your efforts. All of us here really appreciate them.
Hi. I have had lupus for 30 years, and I've just had 2 different types of lymphoma the last 3 years. So I've become very involved in the lymphoma group that I'm working with, and we've been talking a lot about the genome. And the last 10 days, there was 2 articles in the New York Times saying what a bust it has been. It's the 10th anniversary of the finding out about DNA and everything else. The lymphoma group feels that they're very close to finding the genome that is involved in lymphoma. I'm just wondering, I keep on hearing that you've got to have a gene or something to get lupus. Like there's a genetic marker for it.
I'd like to address that. First of all, it's not a bust. It's just extremely complicated.
Right.
And we have to be patient and we have to persevere. But there have been a number of genes identified that are increasing the risk of lupus in subpopulations of people, just blipping up over the top of the background people. They're significant, they're probably real, but probably what's going to end up happening is that it's kind of like you have to have a gene from over here, a gene from over here, but your set won't be identical to the next person's set. And so it'll be an enormously complicated mathematical problem, but it can be solved. With new technologies and new statistical approaches, it can be solved, and it will lead directly to that idea of personalized medicine. We'll be able to characterize a patient, for example, and determine what genetic background put you at more risk for lymphoma, and could we have predicted that things could have been done differently early to prevent that? That's where we're trying to go in medicine. We're not there yet, but that's what this is all about. It's not a bust, it's just very complicated, and we have to be patient, and we have to persevere.
So I keep on hearing that we're doing all of this, research on the B cells, dealing with the B cells. It's going to really take about 30 or 40 years to be actually able to change something in your genetic makeup to that.
Well, hold on a second. When I was a kid, they told me maybe by the time I was 90, we'd get to the moon, and I was 16.
So I guess what I'm saying in the future.
I'm not promising you anything, but I don't want to be that cautious. I don't want to say 30 or 40 years. I want to say let's go to Congress in the United States and what do you have here? A parliament? What do you have here? Parliament. Let's go to Parliament and let's yell and scream and get some money into this issue, because that's what it takes. It takes money and it takes a lot of dedicated people.
Okay, so it's going to be in the future, but do you think that is what's going to be used to help things like lupus instead of,
Yes.
yes?
I'm dominating here. What do you guys think? These are the guys that are going to do the helping. What do they think?
Yes, coming to the genomics and all these questions you have. So you have obviously there are different approaches. One you were suggesting was to identify which are the genes which are predisposing patients to have lupus or lymphoma. That's one approach. The other approach is to try to find out what kind of genetic signature could, I would say, indicate that you will respond better to one treatment than the others. So you have different ways to deal with all of this. And it's true that, as John said, that for lupus, I mean, multiple genes have been identified as being associated with a higher frequency or higher risk of lupus. But I mean, there's still some additional steps to be taken, and there's quite some research going on. But I cannot tell you either how much time it's going to take, but it's moving.
Yeah, it's just interesting for me because I've had the two diseases. I got the lymphoma, and they're taking the approach that of going after the genome of it. And here we're doing the approach of the B cells. So it's just
There's a great deal of work on the genome in lupus, and you know, the idea that this is going to merge into pharmacology is not new. I think everyone's wishing that we were a little further along in genetics so that we could identify the patients in advance who'd be good candidates for the drugs.
well, it's very interesting.
Sorry, even if you can identify, let's say, a couple of genes which are going to predict that you have high risk to get the disease. So that's one step, but this still is not giving you what exactly you can use as a treatment to prevent this to happen. You know, there are further steps to be taken.
Yeah. Yes, he's right about that. The genes are the blueprint for the proteins in your body, and a slight little change in a gene may make a protein a little more overactive or a little more underactive in the job that it has to do in the immune system. So tracking that through and figuring out what's got unbalanced because of that change in the gene is a complicated problem, but the technology is here and people are working on it.
Okay, thanks.
So in our studies, we asked patients whether they would give genetic samples and to look at the genomics, and we have over 1,000 patients agreed to that, and we will do that, but it'll probably take 6 to 12 months and look at things such as interferon signature. All of that has been agreed to, and we will look at it, but it will just be the beginning. Don't expect a definitive answer, but it will certainly add to the database.
Okay, I think we'll call it a day. There's lots going on tonight. There's an opening reception. I hope you'll all be there. And there's going to be a lot to learn here at this Congress. See you there.
Thank you.